Okay, good afternoon, everyone. Kumusta? So I'm Patrick. I'm here to introduce our next speaker for the MSI 50 lecture series. Our next speaker is Dr. Cecilia Conaco. She obtained her BS in molecular biology and biotechnology from the UP Diliman. She had a short stint as research assistant of one of our national scientists, Dr. Luli Cruz. Cecilia went to Stony Brook University in New York for her PhD in molecular and cellular biology. She then had a postdoctoral fellowship at the University of California, Santa Barbara. She returned home as a visiting scientist under the DOST Balik Scientist Program in the Marine Science Institute, University of the Philippines. She was an affiliate assistant professor of the University of the Philippines, Cebu College. She was a deputy director for Bolinao Marine Laboratory, and now she's a full professor at the Marine Science Institute. Cecilia Conaco is a molecular biologist and professor at the University of the Philippines Marine Science Institute. She is fascinated by the weird and wonderful forms of life that thrive in dynamic and sometimes extreme ocean environments. Her research group at the Marine Molecular Biology Laboratory explores the genomic complement of marine animals to gain insights into evolution of animal diversity and complexity and to understand how reef animals make use of their genetic toolkits to sense and respond to the rapidly changing ocean. So, without further ado, let's have Dr. Cecilia Conaco and listen to her talk on stress and sensitivity, what simple animals teach us about coping with change.
Thank you, Patrick. Thank you, everyone, for coming out today. Um, okay. I guess to start, I'd like to. Oh, you can't hear me.
Clear
Right.
audio, ma'am. Thank you.
Right. So, for the benefit of those in the room, I'm gonna hold this thing. All right. Okay. So, good afternoon again. To start, I'd like to ask each of us where we are on this scale, from very calm, neutral, to very stressed. I know where I am at this very moment, but what about you guys? Very calm si Helen. Medyo stressed yung iba? Are any of you in the red zone, you think? Maybe not because it's still the start of the semester. So all of us, we feel, stressed sometimes. But what really is stress? So imagine yourselves as this pile of precariously balanced rocks. Um, this represents the processes in your bodies, in your cells, and in order to maintain a um, functioning individual, a functional cell, you need to keep this balance going. And this balance is what we call homeostasis. It's uh, it's a dynamic process. It ensures that everything works all the time, even when the environment is changing. And stress, as we define it here, is any process that threatens this balance. So it threatens homeostasis, and stress can come in many different forms. Um, but there's a variety of stressors, and it can be different for different organisms. So when you guys feel stressed, what do you normally do? What are the usual? You sleep. You sleep? Yes. What else? What else do you do? You try to. you try to go away, you just try to avoid the stress. So there are many different ways in which we typically respond to stress. So one way is simply just go away from the stress. If you have the means to do so, if you're a mobile organism, that's well and good. You can sleep, or maybe just ignore the stress. Scroll through your YouTube videos, social media, hoping that the stress is going to go away at some point. Sometimes it does. Um, or you can, fight the stress, face it with whatever tools you have, and hopefully you're able to adapt uh, and survive it. Okay, so that's really the goal. Um, so we take all of these different ideas and then we just basically jump into the ocean. Here's a picture of a coral reef. It's deceptively calm, right? It's blue, it's peaceful, the fish are swimming slowly. Um, so it looks deceptively calm, but actually life on the reef is pretty stressful. There's a lot of organisms that are there. There's fierce competition for space, there's predation, there's parasitism, and then there's exposure to all sorts of natural stressors like changes in illumination from the sun, UV rays, changes in the tides, rainfall, which brings fresh water, all of these different things, the changes in the environment that your organisms on the reef feel, all of those can be considered stressors. But there's other things, of course, that add to these natural changes. So we all are familiar with natural stressors, but we also know that our oceans are affected by a lot of different anthropogenic or human-made disturbances like pollution, whether it's eutrophication or plastic pollution or other types of pollution. We have climate change related stressors as well, such as acidification and ocean warming. So these different stressors come in different degrees, and for some of them, our organisms are pretty well prepared because they've experienced these stressors on a regular basis. But are other stressors probably come as a big surprise, and our organisms on the reef have to scramble to respond to these different things. So, what happens inside the cell when stressors arrive? So, here's a cell, here's some stress. What happens? So, we have a whole bunch of different disturbances inside our cells. This can come in the form of DNA damage, unfolded proteins, mitochondrial damage, and assortments of other cellular damage like holes in your cell wall, things like that. Um, but given that stress is a normal day-to-day occurrence and is experienced by all sorts of organisms, we know for a fact that many organisms also have defense mechanisms already in place. So, for example, we know that our cells have repair mechanisms. So, we can repair our DNA. We have heat shock proteins that will refold our proteins if they're unfolded. We produce antioxidants that can protect our molecules from destruction. And then we have ways to eliminate um, proteins or other material that's no longer repairable. So we have processes like autophagy or apoptosis, which work to that effect. So when stress arrives, we can expect a little bit of cellular damage depending on the magnitude of stress. And then the cell deploys all sorts of adaptive responses. If cellular adaptation is successful, then you can restore cellular homeostasis. It's back to normal. Things can function well again. And this is what the process that we term as survival signaling. And for organisms, once you're able to restore cellular homeostasis, you can eventually restore organismal homeostasis. But on the other side, if stress is unmitigated, the stressor is really extreme or prolonged, then cellular adaptation may fail. And in such cases, your cells initiate death signaling. And so in this process, you have mechanisms like regulated cell death or apoptosis and senescence that can be activated, with the idea that if you get rid of terminally damaged cells in your organism, then you can still recover what's still functional. And eventually, maybe this process will also restore organismal homeostasis. So to be able to sense and respond to stress signals is a whole process. I'm presenting here a really oversimplified view of things, but just to give you an idea of what it takes from the stress to a response inside the cell. So first things first, when there's some sort of stressor in the environment, usually you need to have a way to detect that. So you have reception as your first step. This can be through receptors on the cell membrane that will detect some sort of signal. Receptors can also be inside the cell. But the main point is that they are able to detect a change in the environment. And then the next step is signal transduction, wherein from the receptor, you will relay that signal to inside the cell. And this process requires, requires second messengers. So second messengers are essentially signaling proteins, and they can amplify the signal, and they can diversify the signal as well. So at the end of all of this signaling, the relay of signaling, you have a response. And your response can come in the form of activation of certain enzymes or production of structural proteins, all of which are required to maybe maintain cellular structure, repair damage, and things like that. And this production of enzymes and other proteins is usually a rapid response. So you get a change in cellular activity, you immediately address whatever damage has been incurred in the cell. On the other hand, you can also have a response mediated through things like your transcription factors. So transcription factors are proteins that will go into the nucleus, bind to your DNA, and then maybe switch on a gene that you need for the response, or you can also turn off other genes that you may no longer need. So going from the cell surface to the signal transduction pathway, all the way into the nucleus, and then actually turning on a gene takes a little bit of time. So it's a slower response. But what you get is altered gene expression, which is actually important if your stressor is sort of sustained. So it's not going away. I really need to change what genes I switch on because the conditions of the cell have shifted. So there's different options for sensing and responding to stress, but all of these things have to work together harmoniously in order for the organisms to survive all of these changes. All right. So what does stress look like at in the field, in the ocean? So we see organisms in the ocean that have variable responses to stress, just like you guys might feel stressors differently. So for example, if we look at corals, in this picture, you will see some corals that are perfectly happy, but then others that are starting to bleach. So here we see a clear difference. Some corals are more susceptible, some are more tolerant. We see similar things in, say, sponges. So here's a barrel sponge that's perfectly happy, and here's another one that looks a little bleached. And then we also see that in giant clams. So here's a giant clam that's really bleached, but then its neighbors are all just normal and happy clams. So different organisms have different responses. Some exhibit mortality, disease, mortality, while some, just like you guys, are on this particular spectrum where some are able to adapt. Even though you might, for example, some corals might bleach a little bit or get pale, but if the stressor goes away, then they're able to regain, recover, and adapt to the situation, while others are really tolerant of the conditions that they face and remain stable, so no change. The ability to recover and to adapt to stress in the environment is what we call resilience. And I guess there's a lot of interest in looking for organisms that are resilient, especially in this day and age when we know that the oceans are changing and changing very quickly. So differences in resilience, especially on the reef, can cause changes in the community. So for instance, you have a really nice and diverse coral reef, but because you have a stressor, say ocean warming, you might have differences in response of the organisms on that reef. Some will be more successful, while others might not be able to tolerate the stress and die. So what might happen is you'll have a change in the community. So for example, on certain reefs, you might have some species becoming more dominant. So some corals, like Heliopora, tend to be really successful in Bolinao, for example. So reefs can become Heliopora dominated. In certain parts of the world, there are certain reefs that are now more sponge than coral because they have eutrophication and other factors impacting their reef areas. And then more commonly, of course, and in a lot of reefs in the Philippines, we have algal-dominated reef areas. These changes in community also come with changes in functions um, of the ecosystem, right? So, which means that it's really important for us to understand how our reefs are going to change over time, because that will also allow us to predict what functions might change over time. So the question that my lab sometimes likes to explore is what causes these differences in resilience amongst reef organisms. And the way that we study this or look at this particular question is by really exploring the base of the animal tree of life. So we like to study poriferans or sponges in particular, and cnidarians or the corals, mainly because first, they're really common on the reef. So they're reef animals, corals and sponges. Sometimes we also study giant clams, but they're more closely related to us. So they're here in this branch here. The poriferans and the cnidarian groups are also really diverse lineages. So there's so many different types of sponges, for example, and so many different types of cnidarians. So they're inherently interesting in that sense. And because they're early diverging animals, meaning that they split from the rest of other animals very early in, or very a long, long time ago. Sponges, for example, were estimated to have diverged from the last ancestor of animals around 600 to 500 million years ago. So because of this period of separation, they also represent a lot of adaptations to all of the upheavals that Earth's environment has gone through over time. So what we see now these days are modern representatives, and they sometimes, if you look deeply into their genomes, might still hold certain clues as to what changed over time and their history. So we're able to perform comparative analysis, so we can look at what's common, what's different amongst these different organisms, to allow us to get a sense of what traits might be related to what certain genetic sequences. So common traits are usually conserved and very important. That's why most organisms in the modern representatives would still maintain these. While unique traits, which are traits that we might find only in specific lineages or in specific species, for example, um, may be related to specific characteristics that are only found in that particular lineage or genus or species. So depending on how you look, you can sort of explore when these different traits arose and what the genes are that maybe correlate to these particular traits. So we're all very familiar with corals, but I just wanted to introduce you a bit to the poriferans as well. So sponges or the poriferans, they're also, the name derives from the term pore bearers. So poriferans are divided into four big families, and the most species rich are the Demospongiae and the calcareans. So they're a very interesting group to study because they're really quite beautiful. So here's just a few images of some of the sponges that we've encountered on our reefs. They come in different colors, different forms, and it really makes you wonder as to how all of this diversity arose and what's the basis? What is the molecular or genetic basis that corresponds to these different forms that we see? Obviously, we can't directly um, decipher that at this point, but it's an interesting thing to think about. Um, and in terms of stress and stress susceptibility, we can also imagine that a similar thing exists. Different sponges would exhibit differences in their responses to stress. So sponges, as we all know, are also key members of reef ecosystems. So in this particular picture, here's a sponge, and they're important for the cycling of nutrients. They are important habitats and sources of food for certain organisms. And some sponges are also key for reef consolidation or bioerosion. So so we also study these organisms apart from the more popular corals to get a better understanding, of all of the functions that are important to maintain a coral reef as a functioning ecosystem. So how do we study the responses of reef organisms? As a molecular lab, basically we're interested in looking at the genes. The genes, as we know, contain the information that give rise to the phenotype of an organism. So genes are transcribed into RNA transcripts. Then the RNA is read and converted or translated into proteins. And the proteins is what builds the cell and performs all of the functions that are required for a cell to work. So our lab looks here at the RNA level using transcriptomics usually. And this approach, while it has its limitations, does enable us to get a grasp as to what genes an organism has and how it expresses these genes under different conditions. So it's a nice approach for looking at stress responses because we can get snapshots of what genes are on, what genes are off, or how the levels of expression change. So in the next few slides, I'm going to basically show you some of the more interesting things that we have discovered in our work. And I have basically grouped them into life lessons, essentially. Basically, life lessons from our distant cousins, the sponges and the corals. Um, some of these life lessons are going to be a real stretch from the data that I'm going to show you, but Apologies, but that's just, it was crunch time. Okay, so life lessons. Let go of extra baggage, focus on what's important, walk your own path, keep your friends close, and opportunity favors those who are ready. It works for our corals and sponges, and maybe it will also work for you. So, number one, let go of extra baggage. This is exemplified by the finding that sponge genomes tend to be more streamlined. And we think that these streamlined genomes, because there's not a lot of other things that are there, there's not a lot of regulatory regions, there's not a lot of extra genes, it really allows for flexibility, meaning that your sponges have a very spare toolkit, but it enables them to be successful in a wide range of environments. And what you're seeing in this slide is are trees of gene losses on the left and then gene gains on the right. And what you see here is in terms of gene losses, we find that the demosponges, which are shown in blue, appear to have lost more genes relative to the calcarean sponges, which are shown here in yellow. On the other hand, we find that the calcareans had a lot more gene gains over time as compared to their demosponge examples that are included in this tree. So what this tells us is that maybe the core gene complement, the small streamlined genome that's found in these particular demosponges, underlies their success in very different habitats. We know that demosponges have colonized a whole range of different ecosystems. But there are also gene gains, and some of these gains might be lineage-specific innovations that are actually important also, and that contribute to the ability of certain sponges to be happy and to thrive in their unique habitats. So that's how we interpreted this. Obviously, it would be interesting to get a bigger sampling to look at more demosponges, but so far it's looking like the demosponges really do have streamlined genomes relative to their calcarean sisters. Okay, so number two, focus on what's important. And when it comes to stress response, the important things are really those genes that would allow you to protect yourself, to defend against stress, and to repair any damage that occurs. So here what you're seeing is a bubble plot of different genes or protein domains, essentially, but let's just think of them as genes on the right side from different sponge species down here. And then I've grouped them into different functional categories that you can see on the left side. And then the bubble size represents how abundant these particular genes are in specific species. So if you just squint your eyes, you will notice that there's different patterns across the calcarean species, which are these guys here, versus the demosponge species, which are shown in orange down here. So they've focused on maintaining or keeping certain types of gene families. For example, calcareans have a lot of calcium-binding EGF containing proteins, which might relate to cell adhesion, keeping their tissues, tissue integrity intact, also maybe for building their calcareous spicules. And then on the other hand, we have things like scavenger receptors, which are quite abundant in the demosponges. And scavenger receptors, as we know, are important for sensing and recognizing bacteria, and maybe differentiating between food bacteria and symbiotic bacteria. So the demosponges have a lot of those. They also have a lot of other adhesion-related proteins, but different from what was found in the calcareans. And then in terms of apoptosis, the demosponges have an abundance of proteins with the death domain. This means that perhaps apoptosis is something that is more active in the demosponges, or something that they can activate under certain conditions, maybe to get rid of any cells that they no longer need, or if they need to change their morphology somewhat, change their tissue structure somewhat. But if you look down here in terms of defense proteins, like your heat shock proteins, thioredoxin and glutathione, which essentially are related to your redox response, they have almost a similar cohort of these particular genes. So the gene families where you find an abundance of in certain species simply suggest that maybe these families have undergone some sort of functional diversification in these species, meaning that these genes might now have slightly different functions from each other, which make them more flexible in terms of when and where they can act, or how active they can be. So I guess that's one of those important things when it comes to maintaining a response. You need to have many copies of the genes that are important, because if you lose one, it's okay, you have a backup. And you also have the chance for functional diversification, meaning that you can sort of tweak your response a little bit or adjust it as the need arises. The other important thing, or interesting thing, I guess, that we discovered in sponges is that sponges, aside from keeping or maintaining multiple copies of certain genes that they need, they've also borrowed genes from bacteria. So this process is known as horizontal gene transfer. And for sponges, we find that some of these transfers happen quite early in their evolution. So we can see the genes present in a wide range of different sponge species, not just in one lineage. And some of these genes, at least those that we could identify, appeared to be related to things like metabolism or metallopeptidase activity, which could be defense, used for defense, but also for tissue reconstruction, or changing your morphology, shifting your cells around. So we think that these bacterial genes may have contributed to novel metabolism and other potentially unique host properties, although there's really a lot more work to be done to really understand these borrowed genes. Okay, so the third lesson that we learned from sponges and corals, but I'm really going to talk more on sponges here, is to walk your own path. And we see that the sponges walk their own path when it comes to their response to stress. So here I'm showing you, this is a figure that shows you the number of genes that respond to certain types of stressors. And the different stressors are indicated by the colors down below. And these are the sponges that we essentially looked at. So 2 demosponges, and then 1 calcarean sponge, and then different levels of stressors. For Haliclona tubifera, the demosponge here, this is just warming. But for Neopetrosia and Lucetta, this is a combination of warming plus acidification, different levels. It's very clear from this graph that different sponges have different responses, right? So you can see that Neopetrosia tends to have a lower response. Not so many genes change in expression with the stress. But for this particular species, you have a lot of changes in expression. Same with Lucetta. And when we look at what functions are represented in these genes that change in expression, we also find differences as expected. So here you're looking at a heat map of different functions, which I've grouped into defense, apoptosis, and growth and metabolism on the left here. And then again, each of these columns represents a treatment for different sponge species. And then the colors in the heat map represent degree of effect. So blue means not so much, yellow, red means we get a lot of those particular functions affected by the stressor. So what you'll notice very quickly is that in the middle, for Neopetrosia, everything is just blue. So that means that it was not really affected by stress. So the stressors that were applied were not above the tolerance threshold of Neopetrosia. It was able to maintain homeostasis, and so there was really not much happening for this particular sponge. But then you look at the other demo sponge, Haliclona tubifera, you see that there are certain gene functions that were really induced with the treatments, with the heat treatments, I should say. For example, protein folding was really enriched. We see yellow and red here for protein folding, which makes sense because under thermal stress, what you get is protein denaturation, and heat shock proteins are important for refolding all of these damaged proteins. And then you have tissue development and regeneration, so maybe to replace any damaged cells and to grow. And then macromolecule metabolic processes as well are part of the response of this particular sponge species. On the other side, for Lucetta, it has a very different functional pattern of functions that are affected by stress. So you have a lot of response up here for cell redox, homeostasis, and antioxidants. So that says that Lucetta is trying to maybe induce the expression of antioxidant producing proteins, because we know that these are protective responses. Defense response is also enriched, meaning that the sponge is trying to deploy its adaptive responses to try to protect itself or defend itself from the stressors. And then we do see some cell death activity as well. And that suggests that maybe there's already some serious tissue damage in Lucetta, and it may be deploying regulated cell death as a means to get rid of some of these cells. So clearly, we see different responses, different tolerance levels amongst different sponge species. And here we're only looking at 3 examples. So the next lesson we learned is to keep your friends close. So as we know, marine organisms, and in particular sponges, host a diverse array of microbes in their tissues. So these microbes are important for many reasons, and they tend, you can sort of think of them as an extension of the genome of the actual animal, because all of the genes that are represented within the microbes found in the organism also contribute to the functioning of the whole, say, of the whole sponge. So microbes in the sponge can contribute to metabolic complementation, to protection and defense from pathogens, from changes in the environment. And as I already mentioned, they provide an extended functional gene repertoire. So microbiomes in sponges are diverse and species specific. And here I'm showing you just a few examples of sponges that we've worked with, that we've looked at the microbiomes of, and the colors represent different groups of microbes. So you'll notice the patterns look really different for these various species, and the patterns are different from sediment and seawater microbes. So that says that the sponges really must be selecting microbes that they like as friends, that they like to keep in their tissues, or that are beneficial to them in some way. So we see this signature of sponge hosts being able to select for their microbes when we look at the the genes of the host. And actually, you've already seen this data earlier, but just it's presented in some sort of different way. So we know that sponges can select and regulate their associated microbes because they have a lot of receptors, a very diverse repertoire of receptors that are known to be used for recognizing molecules on bacterial cells. So, and these are your scavenger receptors, which are represented here by the yellow bars with the asterisks here. So, as you already know from earlier, Demospongae have a lot of scavenger receptors, and these receptors are very diverse in demosponges. The calcareans don't have as many scavenger receptors as the demosponges, but they do also have those here in the yellow bars. So again, this tells us that um, the sponge host, is able to select for its microbes, and in turn, perhaps the microbes are able to select for other microbes within the community, because we know that microbes can control and communicate with other microbes. So they can produce small molecules, other signaling molecules that might help to control the population of members of the microbiome. And so a balance within the microbial community that's associated with a sponge is important to maintain homeostasis of the whole animal, especially when it comes to stressors. So what happens to these microbiomes when we subject our model sponges to different conditions? So here I'm showing you some of our results where we looked at changes in sponge microbiomes under different types of stress. So here in the first graph, this is Dima sponge subjected to different levels of nutrients, low, medium, and high. And then here, the last two graphs represent warming and acidification and combinations thereof. So these two species, you already saw them earlier. These are the two species, Neopetrosia and Lucetta. One of them we know did not respond to the stressors that we applied, and Lucetta was very stressed with different conditions. So when we look at their microbiomes, we find, for example, that Neopetrosia microbiome was also pretty stable. So no change at all. The patterns tended to overlap even with different treatments. In contrast, if you look at Lucetta chagosensis, we can see that especially with increasing level of stress, we find that the pattern, the community of the microbes was shifting into something totally different. And although it wasn't shifting into something, or the shift was not uniform rather, but what we noticed is that there was a spread in terms of the members of the community. So the communities were diverging from each other with the stress, which is which means that the functions are also diverging from each other. So they're not the same as the normal microbiome, but they're different variants. They're different variants of that particular microbiome with the addition of potential pathogens and opportunist microbes. We see a clearer pattern for this particular sponge subjected to different levels of nutrients. So with low nutrient and medium nutrient, not much changes, but with high nutrients, there's a really obvious shift in the microbiome. So what this tells us is that changes in your microbiome or microbial communities will can reflect an increased abundance of opportunistic or pathogenic bacterial taxa, which can then wreak havoc on the function of your microbiome and its interaction with the host, because now you're maybe losing some of your beneficial bacteria, and instead you have what might be more selfish bacteria that are really out to propagate themselves and disrupt the balance of your original microbiome, and then maybe even cause disease to the host. So pathogenic bacteria. Okay. Okay, so finally, the fifth lesson we learned by looking at by looking at sponges and corals is that opportunity favors those who are ready. And in the context of stress, this means that if you're ready to deal with stress, then you're probably going to be okay. So here, this is for corals. We looked at how coral gene expression changed with thermal stress. And we looked at how they express different stress response genes under normal conditions versus under stressed conditions. And these graphs show you in gray all of the genes, the typical pattern of all of the genes. And then the blue lines represent the stress response gene expression at normal temperature. And the red lines represent stress response genes at high temperature. So what you'll notice is for the top three species, Acropora digitifera, Montipora digitata, and Favites colmani, the expression of stress response genes under normal conditions and under high temperature conditions was pretty similar. So the red and the blue lines overlap here. In contrast, for Ceratopora calyandrum, the pattern is a little bit different. So for under normal temperature, the stress response genes appear to be expressed at lower levels. So you see a skew of the blue line towards the left here. And then under high temperature, the expression goes to the right, meaning that they then upregulate expression of their stress response genes here. So what this suggests is that maybe these three species up here are a bit more prepared to deal with thermal stress compared to Ceratopora calyandrum, which had to switch on a lot of gene expression. And having to do this, having to change gene expression under stress can really be energetically costly for the organism because you're having to transcribe and make new proteins. And it can contribute to poor performance under the conditions that we tested. So, incidentally, I should say that Ceratopora calyandrum was also the only species in this experiment that exhibited bleaching. The other ones were just normal. They were happy. Okay. Another interesting thing that we discovered in corals again is that apart from expressing all of these genes before stress even arrives, is that they also have a mechanism to sequester stress response gene transcripts under normal conditions before stress arrives so that they can maybe mount a really rapid response when conditions become stressful. And they do this through molecules known as microRNAs. So microRNAs are small RNAs, about 22 nucleotides long. They associate with a protein complex that will then sit on your transcript, your mRNA transcript, and prevent translation. So microRNAs with a protein complex sitting on the mRNA prevents translation. And we found that for a specific microRNA that we discovered in a coral, many of the targets that were being repressed by the microRNA actually encoded proteins for repair and damage control. So the transcripts were there, but they were basically being repressed. They were not being translated into protein under normal conditions. But under thermal stress, when we subjected our corals to warmer temperatures, the microRNA is degraded. So it goes away, the protein complex falls off of your transcript, and your transcript now becomes available for protein translation. So you have active translation, and you're able to make your proteins, whether they're repair enzymes or damage control proteins, it helps the coral to then immediately respond to any damage that they might be incurring under the conditions of warmer temperature. So a very rapid response and very robust response can be mediated by microRNAs. So I guess it really just tells you that certain corals are ready to deal with stress. And this is not a surprise because the shallow coral reef environment is always exposed to a lot of stressors. Okay, and so the final example that I wanted to show you simply is that stress response genes are not just expressed under changes in temperature or other stressors, but they can also be expressed during development. So here, this is in giant clams, and what we found is that stress response genes in giant clams are expressed very early in development, in the stage even before they're settling onto the substrate. And this probably helps to ensure survival of these baby giant clams as they're navigating their way through, metamorphosis, settlement and metamorphosis. So, sorry. So the same types of stress response genes are expressed in giant clams, heat shock proteins, hypoxia inducible factor, and other antioxidant response related genes are found very early during giant clam development. So again, in keeping with our life lesson, giant clams are also really prepared to deal with stress of development. So just to summarize these findings, we now have a little bit of an idea of some of the genetic signatures of resilience and susceptibility, at least from the organisms that we've looked at. We know that having a streamlined genome, having an expanded repertoire of stress response genes, gene families, being able to express these genes early and in higher abundance, and then having a stable transcriptome, not having to tinker around with gene expression when conditions arise, and having a diverse and stable microbiome all contribute to stability and ability to adapt to stress. In contrast, of course, the opposite of this contributes to mortality. So if you have fewer stress response genes, you don't express them at the level that's required, and you have to then switch on genes during when you have certain perturbations. And if you have a less diverse microbiome that might be more susceptible to intrusion by pathogenic and opportunistic microbes, then you might not have as much of an advantage when it comes to dealing with stress. Of course, it's important to note that these findings really are based on a very small sampling of the reef animals that exist, and that there are many more that are just waiting to be explored. But the more we understand these different organisms, the better we will be able to predict what will happen on the reef, and maybe even make better decisions about how to take care of these ecosystems. So more studies required. Anyway, so what next? Just a few teasers on what's next. So how do we take this data on genes and responses to stress and then extend that? So one of the ways that we've been expanding is actually by going deeper. So here we're looking at adaptations in the mesophotic. So most of the studies we've looked at are shallow reef, coral reef organisms, very exposed to stressors. But what about in the mesophotic, where it's maybe a little dimmer, a little cooler? How do organisms here respond to their unique environment? And are they still capable of adapting to, say, if they were brought to a shallow area? I mean, if their environment, say, warmed or became more illuminated. So different stressors might again impact organisms that are really used to a certain habitat, and they might respond in a different way. Another thing that we've been doing is using information on resilience and the genes that are signatures of resilience, maybe to select for stress-tolerant corals in this case. So you can breed corals and their symbionts that have naturally higher tolerance. And the hope, of course, is that you'll be able to put this out on the reef, and they would then have a boosted response to, especially to warming seawater. So this is through our ACR, ongoing ACR project. Another thing we've been doing there is exploring the microbes that are associated with corals and looking at whether they might be, if we can use this information on the microbes to see what's beneficial to the corals, and whether if we add these back, they would then enhance survival and maybe tolerance of our the corals that are cultured. So a lot of applications for this particular supplements essentially, but it requires knowledge of what the beneficial microbes are, and even what type of nutrition you can provide to your corals, or say to your clams, to enhance their survival in culture. And then lastly, we're also quite excited to be able to then use all of that data on transcriptome, what genes change, to then be able to look for genes and possibly promoters of genes that might boost performance under stress. So can you take these protective genes, put them in, say, a coral, and then have it be a gene that's inducible under stress conditions? Would that allow your coral to have superpowers, so to speak? Would that boost survival of our corals? Of course, this is a new project, and it's really more for developing the technique to be able to do this, because right now there's no way where you can play around with corals, coral genes. Okay, anyway, so many thanks to my home at MSI for allowing me to tinker around with all sorts of organisms. UP for the funding, for getting started with all of the sponge work. Mom Giselle is here, so obviously to acknowledge Mom Giselle for supporting the first project I had at UP with the Balik Ph.D. program. And then, of course, DOST Picard is one of the major supporters of some of this work. ACR, the International Foundation of Science, and just BFAR for help for allowing us to collect things on the reef. Finally, of course, acknowledgements to the people who actually did the work. So I've been really, I guess, fortunate to have worked with such an exemplary set of students. So most of the sponge work I've talked about today were done by Tin Guzman, Nino, Jake, and Mike. And then the choral work was done by Adi and JK. And then our giant clam mother is Kiana. So the data that I showed you, they generated those. And then, of course, I have my other current team from the MCE, the Mesophotic group, and my choral group at BML, who are, well, hopefully tirelessly working towards understanding corals and sponges a little bit better. So thank you so much, and I would be happy to answer any questions.
Thank you very much, Cecilia.
Hi, Cecile. Thank you for that fascinating talk. Just curious, I'm sure you've come across recent developments about coral reefs. You know, the papers that are showing that there are so many lineages of what we used to think to be a few or even the hundreds multiplied by 20 or 30 more because people are now looking at lineages. So I'm curious, to what extent are these patterns, because it's species, diba, specific, even the corals, might I mean, well, of course, you were looking at particular colonies. No? So maybe there are other cryptic lineages, many cryptic lineages. How would you imagine this to be? You know, will it multiply all the diversity, or
Mm, That's a really interesting question, and actually something that sort of connects to what we're doing because we've been looking at corals on the same reef
yeah,
and um, and we're able to categorize them by tolerance levels essentially. So given that new paper where they talk about all of this cryptic diversity, it's also possible that the ones that might be high tolerance might be different really genetically from the ones that are low tolerance, but that would require a lot more really analysis. So it would be very interesting to actually do that on our reefs here. Clearly, there's diversity that we're unable
yeah.
to handle on at this level, but there's probably a lot more nuance to really being able to distinguish different lineages, especially I think for the acroporids, which are so I think so diverse.
Yeah, so maybe, you know, in future you can have the other people do the lineages together with what you're doing. So we
Yes,
have a more integrated, kasi
more, yeah.
kasi you have the mesophotic and the shallower, no?
Right.
So it's really a fantastic opportunity to put different studies now together, no? So thank you for that very exciting work.
Thank you, Ma'am Annette.
Okay, Ma'am Annette, any more questions?
Hi, ma'am. Congratulations on your talk. I really love the life lessons. Um, Under global change scenario, like for example, thermal stress, certainly there will be winners and losers. But I want to know your insights regarding the stress response of sponges and other marine organisms covering latitudinal gradients from polar, temperate, and tropical environments. Would there be a significant differences on the stress response and the level of susceptibility and resilience as reflected on their genetic signatures?
Thank you for that question. It's an interesting question, but I think we can expect that, given the exposure history of these different communities or populations of organisms, that they would be used to a certain level or of say temperature and level of fluctuation of these conditions in their environment. So I would imagine that we would expect to see differences in tolerance levels. And I think when you look at papers from other countries, they do use different say temperature as a stressor. So for us, that's probably this level is probably not stressful because then our corals do experience much more extreme conditions. So so yes, and that's really interesting because you can then cross compare across groups of organisms in different, say, latitudes or areas, and then see whether there's any commonalities in the types of genes that they might have, whether they have expanded gene families for certain things, because this is, how they then use it to respond to a particular stressor. But yeah, I guess the short answer is yes, we do expect to see variation, and it's just how complex it is.
Okay. Go ahead, Ma'am Lau.
So ano muna, may tanong ako kanina pero sesegundahan ko lang yung tanong ng latitude. So therefore, ano consequence nito sa pag-rear ng culture for recovery, right? So does that mean at that young of, you should start stressing them out so that they can express early. So by the time you put them out there, they're used to it. Or, I mean, at what stage, or does that mean also if you don't stress them out, that they will have less or they'll have a harder time in surviving? Nanay to, nag-iisip ako sa anak. Right? So,
Well, it's a really interesting. That's a really interesting question, and people are doing experiments like this. Para siya, I guess they call it assisted evolution, where they would rear successive generations with higher and higher levels of stress, in order to try to reach a particular tolerance level. And you can do that, yes, with the larvae that you have, and just gradually build up their tolerance. The other approach, of course, is to find naturally occurring populations of these organisms of interest that are already parang hiyang, or used to a certain, stress level. And maybe read those, and they would already be somewhat more equipped, and you can then further assist the selection by the breeding of different individuals. But
thanks for that. Can I have another question? So, tanong ko lang, is there a way of measuring the how expensive energy-wise is and compare the existence of your microRNA that suppress certain translation versus having already stress response gene
like
that's
already on.
yeah already on.
I think you can, because you can sort of, how much, say, ATP is needed to transcribe and then make proteins to translate. So you can get that computation done. Also, if you think about it, when you're under stress, you're still having to keep up with your metabolic processes, right? So having an additional thing that you need to like make new biomolecules is definitely, you can imagine, it's definitely going to be an added parang burden on the cell. I have to make new proteins, and that's going to take away from ATP that I need to simply survive this particular time in here.
Okay. Thank you.
We have a question from Sir Endo.
Kung okay lang, may magkakadugtong akong tanong. Sa mga experiments, una, ano yung variables sa mga temperature, and then you have nutrients, and then you also have yung sa acidification. Ano yung increments of change na ginawa ninyo? Pangalawa, gaano katagal na in-expose yung mga alaga mo bago na express nila yung response doon sa experiment. So, one. Two, related to that, if you are to think of the Philippines latitudinally, the question asked earlier, temperature-wise, can we have some sort of a variation in temperature that is sufficient para magkaroon ng pagkakaiba sa gene expression ng response ng organisms? Okay. Then certainly, you know that there are environments where if you want to look at warmer temperatures, either you go on the reef flat, which is very different from the reef front, or you go to Mabini, which is also much warmer than other places. So sa mga ganoon, yung results ng trabaho nyo, ano yung potential na relationship niya doon sa mga natural na environments na variable, yung potential na levels nung stressors na tiningnan mo or tiningnan ninyo?
All right. Thank you, Sir Ando. So let's see if I remember. Rate of change and level of stressor. So iba-iba. But for the more recent experiments, so before we were doing really acute exposures, as in we just put the organism directly into hot water. But in later experiments, we've adjusted this where we can now ramp, slowly go up to the level that we need. And then we expose them not so long. So we can run experiments typically ninyo a week, about a week. So really short-term experiments, rapid ramping, which will in no way approximate what's in nature. But we do this because then you get a sort of what you want to get is sort of a handle on tolerance limits, and also what genes would they actually then deploy under these conditions. And our assumption is that, although that remains to be really tested empirically, is that these organisms are going to use the same genes to respond to similar conditions on the reef, which are probably going to be less extreme than what we applied in the experiments. Because on the reef you have, up and down, di naman sustained. So in our experiments, they're really they're really in the condition for extended periods of time. So it can be a little more extreme for the organism. And in terms of being able to look at different parts of the Philippines, so yes, it's a very interesting sort of laboratory, I guess, for Philippine scientists, because you do have areas where you would get very cooler, maybe ocean temperatures, versus areas where you would then experience really high temperatures, especially say during summer, or if you have El Nino and things like that. And it would be super nice to be able to look at differences in response across these organisms. And that, I think, would be something to look forward to in the future. There's great interest in being able to see how similar the gene repertoire it would be for organisms that are used to a particular condition versus, say, a more moderate condition versus, say, ones that are in more extreme conditions. And I think maybe there's a lot of there's still a lot of things we don't know, I should say, but we have a lot of students who could possibly look at all of these different areas across the Philippines. So yeah, so I don't know the answer because I haven't really looked. We've only focused, a lot of our work is in Bolinao, which tends to be really hot, which is good for some of our selective breeding studies. But there are people who look more broadly across the Philippines. And I think while stress response genes tend to be conserved because every organism has these genes, what really differs is in the way that they regulate the expression of these genes, and maybe even how big these families are in each of these different groups of organisms. So that's something we don't yet know, but would be really interesting to explore, say, in the future. Did I answer your question?
So certainly madaming pakinabang, madaming gamit ang lab work. Pero the Philippines is a natural laboratory.
Yes,
And
for
I
sure.
would encourage students to go out and look for such topics. There's so much variability If you want 40 degrees daytime and then nighttime, bababa siya ng 24 or even lower, go to a reflat. I've based in a tidal pool in a reflat na halos 40 degrees centigrade ang tubig sa hapon. And that takes place every day. Okay? You can go to an area with shallow hydrothermal vents, but you have all of these organisms nearby. So all year round, the temperature is higher, the pH is lower than normal, plus a lot of other things. And I think ang daming potential discoveries for the different fields no dito sa MSI at maganda na people work together kasi mahirap naman na sa dalawang lab ay matingnan lahat.
Thank you, Sir Ando.
Okay, more questions? Right. Michelle?
Hi. Hi, Cecilia. Congrats. I have a couple comments or discussion points. Una, si Lucetta chagosensis, aware ka ba na species complex din siya? Aside from what Annette was saying about corals, Lucetta, there's already some data, pero wala sa Philippines, as far as I know, na species complex siya. So that's also something worth checking. Pangalawa, and I was thinking of this dahil dun sa interesting comment about latitudinal gradients. Another hypothesis I have na I think it's untested is, well, first, I have this hypothesis that species that have a narrow latitudinal range, that have a small geographic range, iba yung kanilang life history, whether reproduction or stress response, to those whose range can range from subtropical to tropical. And I thought it might be interesting to say compare a species of sponge, two species from the same genus, na isa narrow ang range at narrow ang thermal or whatever tolerance versus something that's very wide-ranging. And then tingnan both yung transcriptome and yung mga gene duplication. Anyway, so just going beyond, parang adding to the discussion, if we expand it, Philippines. But I think that's still too narrow given marine species are, they have huge ranges. I think ang mas interesting palalo if we collaborate with institutes from, say, subtropical areas, and we compare the same species and we're sure they're the same biological species. Yun lang. Discussion points lang.
Thanks, Mash. Yeah, so for sure, I think really one of the, so yes, I agree. Very interesting prospects for future studies for students to think about. But I think to get there, we also need to know our reefs better, like what's here. Is this the same species as that species? So,
Yun yung una.
so yeah, that's the first thing really. And then to do the comparisons, which would be really cool. Yeah, thank you for that.
Hi, Cecil. Yeah, thanks for that talk. Wala ko alam tungkol dito eh, so I might be talking about something that is well known. But one thought that came to mind was the talk seemed to emphasize individual variation in gene expression in the beginning. You were asking us how do we feel, diba? And then, but a lot of the variation at the species or higher, taxonomic levels were also emphasized. Given that, it's not looking good for things like corals, no, in the next 25 years, 30 years, for example, thermal stress, no. Do you think that there's a choice there? Where should you devote much of the attention sa research? Is it going to be in questions that relate more to individual variation or
the types of levels of variation that you were talking about. And that probably has something to do with those two previous questions, population level, geographic distribution. So what do you think about that? Is that but with that, you can also think of, I note that in the ACR project, for example, may technologies that can come from breeding, for example. But do you think there's a priority that needs to be addressed as well? Thanks.
Not quite sure. I guess from my perspective, kasi you do look at, it's one. You treat each one as an individual, and then you sort of extrapolate to how it would work as a population. So it's tough for me to say whether you should focus on this inter-individual or individual differences versus population, but I guess all I can really say is that we need more information on a lot more representatives, because we tend, even for the current work that we're doing, it's, you're really looking at certain organisms of interest, your most favorite model, and there's, as Michelle also mentioned, there's really a lot more diversity out there that remains maybe unexplored. So yeah, but yeah, so it's also important to note na, yeah, so you have inter-individual differences, and then you also have the species differences, and we also do see that because you get a grasp of, so sa end, iba-iba talaga yung tolerance limits ng iba-ibang species, but you do see a lot of variation even at the individual level. So the resolution of how we can look at it will be limited siguro by, the approach and the cost of the approaches that you use, where you can't really do a whole, I don't know, like 100 transcriptomes in one. So, but yeah, but I think, yeah, so you knew. So I probably I did not answer your question, but
Kung may future students, remote sensing can also provide historical spreads. So, pwede namin masabi sa'yo saan area ang nagkaroon ng heat stress, ang mula ng sobra, so that we want to see kung iba yung response ng historical stress versus those for things.
right.
And there's different granularity with the scale, some
Interesting.
Just a quick comment and then a question. Cecilia, what you presented is actually a toolkit that can be applied to any organism on the planet, right? Whatever the level of complexity, whether it's a simple alga or even simpler, up to the mammals. So all of them have genes and express those genes in the process of adaptation and eventually evolution. So I would just like to suggest that the perspective can be much expanded exponentially to look at life on the planet and how it's adapting, responding to potential stressors. Speaking of which is my question now. So what you presented are results of millions, if not hundreds of millions of years of evolution. But the anthropogenic signature of abuse is just so huge. I'd really like to know your personal opinion as to how this is gonna affect life on Earth. And I'm not just talking about natural stressors like temperature or so-called greenhouse gas effect. But all of the pollutants were dumping not only into the ocean, but on land. And chemists, with all due respect to them, chemists, you are inventing all kinds of compounds all the time, right? In the name of improving human life. But you are not presenting solutions as to how to deal with the unforeseen effects of these compounds. I mean, not to mention plastics. So they're just accumulating and accumulating on the planet. And how are these mechanisms of gene transcription, expression, translation, how are they keeping up with all of this human caused abuse? And what is the outlook for the life on the planet, even within the century? Thank you.
Thanks, Helen. Well, so as I mentioned, while the mechanisms for responding to stressors are conserved and have evolved over many millions of years, it's likely that just the scale at which these stressors are now affecting organisms might be too much for the mechanisms to adapt uh, and change accordingly. So perhaps I think for many of these organisms, the levels at which we're dumping all of these things or the stressors that they're experiencing um, might be sort of really noxious to them. So the conditions are extreme. The mechanisms of the cell cannot keep up. And so the danger really is that even before we've explored these organisms and how they can deal with stress, they're already gone, right? So, but that's because of the way that evolution works. It needs a lot of time to really to get to an organism that would be able to survive. But who knows? Who knows? There have been a lot of upheavals, right, in in Earth's history, and they're still here after 600 millions of years. Yeah. Oh, right, right. So
Try to perceive what life on Earth
would look like. Yeah. I mean, yeah, that's something to think about. But I guess really the main thing is the stressors are the degree of the stress is really just extreme, and they're patong-patong. So organisms, I guess, would be scrambling to deal with all of these things, and maybe some of the stressors are not things you can anymore deal with the usual adaptive processes that you have because pagtatabunan ka na ng plastic. Well, that's the end of you. But but I don't want to be too pessimistic, but yeah.
Hi, Ma'am Cecile. We'll be reading questions from Zoom and Facebook Live. So we have one from Zaldi Mabolok. Hi, Dr. Conaco. Great talk. I have a general and a more specific question. Epigenetic modifications and transgenerational effects in other groups have been shown to help organisms adapt to environmental changes. Any similar studies or observations in corals and sponges?
Was that the question?
Yes.
Okay, so thanks, Zaldi. Yes, there have been a bunch of studies looking at epigenetic changes in corals experiencing stress. Although the outcomes of this, like how it actually affects gene expression and what specific genes are being affected by changes in epigenetic modification, I think remains to be further explored. So basically, people are looking at epigenetic changes, methylation in particular, as a whole, and future studies, I think, would then start to really pinpoint where in the genome these changes are happening, and then what the outcome would be, and whether how it relates to the stress response.
And then he also has another question. So in one of your studies among coral species, Ceratopora calliandrum, being a brooder, exhibited bleaching and more stress. Do you think the reproductive strategies of the corals, breeders versus broadcaster, affect their stress responses?
Possibly, yes. Brooders, you would expect they, as Michelle already mentioned earlier, they would have more narrow ranges. They would settle very close to their parents, and so their area, their habitat where they're exposed might tend to be limited. So the conditions would also be maybe more stable. So probably they're less used to really bigger shifts in their environment, and as such, maybe the stressor that was provided to them was really more than they could handle, and so they had lower tolerance overall. So yeah, it's possible, and it would be really interesting to do more comparisons in the future.
And we also have a raised hand from Maria Michelle Ann Perez from Zoom. Okay, maybe we can go back to her once she unmutes.
Okay, any more questions?
I know, very nice talk. I have a very simple question. In terms of conducting your thermal experiments or other stressors, it's a common practice after getting your organism from the wild to your laboratory or whatever setup. You do a prior acclimatization to the organism. Would the acclimatization period affect the transcriptome of the organism once it is under the actual treatment, or would it make no difference maybe for some organisms?
Yes, it would affect the expression, because the transcriptome is very dynamic. It responds to, all of these changes. You cut it, you put it in a totally different environment from where you got the organism. So yes, and this is why we do controls where you have the same organism also experiencing all of these additional stress before you do the thing. But whether it affects the actual response, we're not. Maybe. If you don't give it enough acclimatization time, then it's sort of compromised as well, because it's still healing. So that could also be a factor that would affect what we see. Yeah, so something to consider, obviously, when you're analyzing the data.
Thank you.
Thank you. Si Ma'am Giselle.
Hello, can you hear me?
Yeah.
Congratulations, Cecilia. It's like I was reading the book that I use as a major reference when I last taught marine biochemistry. So what you covered today is very important for evolution to be understood at the various levels of, molecular control and lack of control. And well, from a philosophical standpoint, which I'm sure Helen Yap loves. Where is Helen? Is she still here or she's gone? Okay.
She's
Yeah. So it's really about control and over time and space. And well, anthropogenic activities and technology are part of evolution. So live with it. So anyone who makes use of any technologies to further knowledge should learn to live with it. Plastics are not completely bad. So there are many uses of plastics. So it's good to move towards multiple use plastics, not one time use. There are many things that we cannot do without polymers, and that's really a product of human knowledge. For example, if you're sick or you have a relative who is sick with cancer, you need all the plastics that are found in hospitals, right? Anyways, and so with the drugs. But here at the Marine Science Institute, we study drugs that are natural. Of course, we can do chemical modifications and they can become synthetic. And then, we throw them away and they're cytotoxic. We don't put them away properly, so that could have impacts. But anyway, I think that the order of the day is to deal with climate change and rising temperatures due to GHS, to greenhouse gases. And it's been a long time that we've been thinking about this at MSI. And now there is like a proposal to just do carbon capture, carbon dioxide capture, not just in land, but again also in the oceans, when you know that in fact the oceans already capture a lot of the carbon dioxide. And I will always remember what Jacinto, my teacher in chemical oceanography, would say, that, then we would have the acidification, right? So, and yet there are people, you're right, there are chemists, chemical engineers who will model this and say, oh, come on, let the oceans, capture more of the carbon dioxide, okay? Not knowing what the consequences would be. Anyway, I think that MSI should continue to think about these things because we are in the era of innovations that are actually a response to the world that we cannot control. So there are many uncontrollable factors, and we just have to learn to adapt to things that we really cannot control, and that's the nature of evolution. And the biological equivalent of that is homeostasis. We know it in biochem, right? The cellular level or the organismic tissue and organismic level. And there are many factors that come into play. So I think that adaptation or adaptability of cells, of organs, tissues, humans, and ecosystems follow the same principles of competition, cooperation, and adaptation before the next deluge or the next, annihilation. And if you want to read about this, there's a book about it. Everything, the periods of annihilation. I don't know if Lilibet has shared the book with you, or Isa has shared the book with you. It's a very good book, okay, with all of the transcriptomic analysis as well of marine organisms. But I'd like to just also think about how your study relates to the marine biotech or the natural products group, because they're not here. Lilibet is not here. Isa is not here. Roy Luisma is not here. Where are they? Okay. So I guess I have to ask some questions. So I'm just curious in your studies in terms of control. So it's nice strategy of evolution to have controls, regulation at different time scales and space scales, and there's always a cascading or amplification effect. So I'm just wondering how you would view microRNA regulation versus interfering siRNA in terms of control. And I used to have the view that everything is about specificity. Everything is about specific interactions until we learned more about transcription factors and RNA mechanisms, which are not necessarily specific. In fact, they are more general and they're more a synchronized regulation. So I wanted to get your views on this and how major these mechanisms are relative to one another, how they compare. Also, whether the very revolutionary mechanism is found in microorganisms, this is gene editing or cluster, is existing or happening to a large extent in the demospongi, the cnidarians, and all your organisms of interest. Okay. And finally, meron pa. Okay, meron pa. Baba ng pababa hanggang sa my interest, for example, in cancer, no? Or inflammation. So we have a publication on a compound. It's called boholamide. And the way this was discovered was through our DRG assay. It's an assay. It's a cytotoxicity assay of glial cells, neuronal cells. But then these were put in hypoxic environment. Okay, so hypoxia-inducing factors are produced, inducing or inhibiting factors are produced by organisms. This compound was found in bacteria, and so I'm just wondering whether this is really a major area of study because of reduced oxygen, hypoxia, okay, on the one hand, and also lots of oxygen, activated oxygen or reactive oxygen species, which are found in the mitochondria during the oxidative phosphorylation process, right? So, in fact, a lot of the harmful oxygen is not just because of the external UV hitting the oxygen in the air. It's really because of the loss that is produced during metabolism. Okay, too much activity in metabolism. So I'm just wondering whether anybody or you would try to lead research in this area, because in cancer, you really have a hypoxic environment. Well, cancer is caused by ROS, damaging ROS and RNS, reactive nitrogen species, reactive oxygen species. But then cancer cells have adapted so that they can live with low oxygen. They are proliferating so fast, and the surrounding oxygen is not enough, and yet they have mutated to survive. So, there could be models in these stressed environments of low oxygen to find compounds that could, be effective against cancer, just like boholamide. Okay, so I'm just saying that I know we're not in control of a lot of things, in our lifetime or even in our the generation, but during this period of our existence, I think we should try to help, improve the health and security of humans and the planet. So that's one. The other final is symbiosis. So you talked about the sponges actually borrowing from their inhabitants, their friends, their little friends, the microbes. Okay, but in fact, there are these microbes, like the blue sponge that hosts a microbe, for which our former RA here, Diarrhea Tiamero, published as a first author in Princeton about how this bacterium in the blue sponge that's been cultured by Vanessa and Perry and Lilibeth, okay, produces the compound of interest, a very cytotoxic compound, but you can no longer isolate the bacterium because it has really lost independence genes. So it's entirely reliant on the blue spot. So it's the real, symbiosis that you could define as not being able to exist one without the other. So, anyways, I'm just curious if you're also studying sponge symbiosis very, very closely. Okay, thank you.
Thank you, Ma'am Giselle. Um, yeah, so lots of questions. Sige, I'll try, ma'am. Um, so regarding the question on microRNAs as a regulator of the stress response versus the siRNAs. No? So I think one of the general ideas is with microRNAs, you are able to get that repression, but maybe not the degradation of the transcripts. So it does allow you to hold on to many transcripts and just keep them sort of quiet for a while until you're ready to use them. So perhaps maybe that's one big advantage of having the microRNAs as a mechanism for stress response. As you also pointed out, microRNAs have they can bind to many different transcripts, which means they can actually control a lot of different gene expression, yeah, all at the same time. So that also gives them a lot more power to regulate anything maybe that's say related to stress. So just one microRNA, many different genes, and then you can remove that control immediately with thermal stress and have all of your genes expressed. So it's a really attractive mechanism. I'm not really sure if it's really a universal thing, but they've shown similar things in other organisms. Under stress, you do have loss of microRNA control on certain genes, and it's important for the response. In terms of editing, RNA editing, for example, although it would be a really interesting thing to look at. There's not any reports yet in sponges whether it's at work. It's more commonly known, I guess, in mollusks. But I wonder if it also does play a big role because it's also important for generating diversity in the transcripts, right? And just giving a lot more maybe refinement to the activity of the proteins that are made. So that would be a nice thing to see. But we haven't done any work on that yet. And then in terms of hypoxia, yeah, that's a really interesting idea. And there are studies, for example, that say that there are a lot of micro niches in sponges that form, say, when they get covered by sediment, and so you do have hypoxic pockets, and they know that there are microbes that are really specific for those habitats, micro habitats within the sponge. So if that's something that could be replicated in culture with your blue sponge and the microorganism that produces that particular compound, that would be really cool, right? So you have a little microcosm type thing. Yeah, but we haven't done much work really on that, although we have explored transcription factors in general in sponges. So Nino has done that, and I'm not sure if he's recovered hypoxia inducible factor in some of our species. But yes, so we do look at different factors, and we haven't yet teased out like where and when they would be expressed within our organisms. Yeah. And finally, maybe just also to address the thing about evolution and all of the changes. I guess as they say, diba, the only constant thing is change. So that's really so we're all just evolving as we go along. But Mother Nature is amazing, right? So she's come up with so many different solutions to the same problem, like having different morphotypes, having different phenotypes, and all of that. So the combinations of genes and the way that we use these genes could really come up with a totally different, thing that might then be suited to whatever conditions are being selected for. So it's absolutely possible given within the realm of, how we understand evolution, I guess. Other questions? Oh, I see a hand up.
More questions?
Thank you for the talk. Not having background in any of this, I just wanted a bit of so as I understand, there's stress, and then because of the stress, the cell will react. And like what you showed earlier, different organisms react differently. But then I guess I can understand that, and like, okay, this organism is not reacting, meaning they're still okay. But how does it translate to surviving? Because I can imagine that maybe a certain organism will react because, oh, this is the stress I'm feeling, this is how I'll try to like address it. But does it translate to actual like being able to survive and like persist, I guess? Uh yeah.
Okay. A very existential question. How does the change in the way that you express your genes contribute to survival? That's how I understood because yeah. Well, we won't necessarily know it will survive, although we know that it has the ingredients it needs or the genes that it needs to survive. Um, but we can see that it's trying, right? So when you deploy the different genes, you know what they do. So you know that this gene is going to repair my DNA. This gene is going to get rid of this particular damaged protein, things like that. And being able to keep your cells in good condition through these mechanisms is what eventually will allow yourself and then the organism itself to survive. But just because you express those things does not necessarily mean you're going to survive, because if the stressor is more than your cells can deal with, like the damage is too much, there's just too much damage to deal with, then maybe it's still not going to survive, but it tried. Diba? So, yeah. So that's really all that it tells us.
Any more questions?
Kaya nga,
From Zoom, wala na? Wala na? 1, 2, wala na? Okay.
If you have other questions, my email is up here. We will try to address any. Kung di kayo makatulong, tapos may tanong kayo about stress, yeah. If you're a sponge, yes.
Okay, thank you everyone. So we'll close this seminar. Thank you, Cecilia. So if you have questions, you can approach her or email her. Okay, we have snacks, cafeteria. Thank you.